guess we'll get started. Thank you everyone for coming. It's a very special seminar we have today, and it is great pleasure for me to welcome my old friend, Dr. Vitaly Dunin-Barkowski and his group. I know Vitaly since I was an undergraduate student attending a similar seminar at the Research Institute in Russia, and I was very much impressed by contribution which Vitaly was regularly making through this seminar series, often presenting provoking and always interesting topics, subjects. Vitaly had worked over 10 years in the United States in various positions, and during this time he even managed to talk me into a collaborative research, which in addition to telling me that he is indeed very interesting person to work with, also showed me that the research that Vitaly does is extremely focused and going very deep, which kind of convinced me to invite him for the talk today when he called me and about a month ago telling me, Irina, I'm not crazy, but I want to present to you a crazy idea. Would you let me do it at Barrows? And knowing that he is a very deeply researching person, I said, sure, of course, welcome. So, and today we have two 25-minute presentations by younger colleagues of Dr. Dunin-Barkowski. Following each presentation will be followed by 5 minutes question-asking session. After that, we'll have coffee break with brownies and cookies, 1:15 through 1:30. We'll have coffee break here in the lobby. And then everyone who truly wants to sample Russian computer brain artificial intelligence, is it there? So welcome back. Where we will be treated to 40 to 15 minute presentation.
It's not artificial.
No. Okay. So when Dr. Vitaly Barkowski will talk himself, and during this session, I encourage you people to really interrupt him and discuss whatever you are going to hear, because I warn you, some of this will be controversial, I think. So now, the first talk in this series is the one by Yakov Karandashov, which is entitled "How many neurons can make many of grandma neurons?" Please.
Thank you very much. I would like, firstly, to thank Irina Belovezova for invitation in this very interesting institute. I was impressed, and I'm glad to report my presentation, how many neurons can make many grandma neurons. My name is Yakov. Let's start. Among neuroscientists and neurophysiologists for a long time, there was a hypothesis that there must exist some neurons that represent an object or concept, and firstly in 1967, Tsikhanovsky suggests the concept of gnostic neuron, and 2 years after it was called by Jerry Latrin grandma neuron. That is, the grandma neuron is a neuron that represents a specific concept or some object or maybe person. And recently, in 2005, Quiroga, Koch, and et al. Show that they found in medial temporal lobe some neurons, such that neurons of Jennifer Aniston, neurons of Bill Clinton, Halle Berry, and etc. And what question that arises here is the first question is that the grandma cell is not cell. It's a population of neurons, many neurons. And at the same time, because there are a finite number of neurons in the brain, some of neurons must be involved in different grandma representations. And here we would like to present a model which satisfied both conditions and looks very attractive. And I will explain it here. The idea is based on the attractor neural networks. Donald Hebb, based on the ideas of conditional and unconditional reflexes suggested by Ivan Pavlov, proposed a hypothesis that the presynaptic and postsynaptic, if the presynaptic and postsynaptic neurons are activated simultaneously, then the existing synapses between them will strengthen. And based on this hypothesis, David Marr in 1971 and after that John Hopfield in 1982 proposed construction of associative neural network, which I would like to illustrate here. Let's suppose we have a population of neurons, and some subset of these neurons are active. Then, according to the Hebb hypothesis, the connections between these neurons will strengthen. And this means that such network of neurons is memorized this pattern of activity. And later, this pattern can be retrieved by a small part. And if we repeat this procedure several times with different patterns of activity, all of them will be learned. And each of these patterns will be the attractors of the network, which means that if we start from a pattern of activity which is close to one of them, then the activity of the network will go to this pattern. And the main idea that we proposed in this work is that really the grandma cells are such attractors in the brain, and we found another way for constructing such attractor neural networks with the model molecular markers. What is the model? Let's suppose we have m different markers. I denoted them by different colors. And let's suppose that each neuron from its birth have a k different markers in it. here k is equal to. And let the neurons that have the same marker will create connections. That means that if during the axon growth process, if one neuron meets another neuron with the same marker, then the synapse is created. In such a way, the neurons, L neurons with the same marker, here L is equal to 5, markers, red markers, they have, all of them have the same red markers. All these neurons with the same markers will be interconnected. That is, they will form the attractor of this network. Analogously, the neurons with another markers, here is with the green, will again form another attractor, and so on. Because we have m different markers, then in the network will be inborn m different attractors in which of L neurons, and because each neuron have k different markers in it, then each neuron is involved in k different attractors. And what is the markers? You can consider markers as pieces of DNA or some special proteins. I will not stop on this question. And when this process is finished, we have the network of neurons with preformed attractors. And we investigated the question about how many attractors can be obtained with such molecular markers, and found that the maximal number of attractors that can be obtained is quadratically increases quadratically with the number of neurons. And for example, if we consider cortical column as a basic elementary neural networks with n with the number of neurons equals to 50,000, and with the average number of active neurons about 50, then we can obtain up to 1 million different attractors. That is, in the network of 50,000 of neurons, we can it can be created up to 1 million different grandma representations, which is about 20 times larger than the number of neurons. And the final question is when the main idea that we propose is that there are some inborn structures in the brain. These structures are attractors, and they are really preformed. And during the life, they absorb the meanings. That is, the real concepts or objects or persons are involved in these attractors only during the life. and how it can be really. Let's consider the simple scheme of Rosenblatt perceptron. What is Rosenblatt perceptron? It is such structure where an input layer of neurons, output layer of neurons, and the connections between the input and output. And what does it do? Suppose we have a set of input vectors x and some output target vectors. And we would like to teach the perceptron to give the target vector in each input vector. It can be done, it's usually done with the Rosenblatt rule, and which you can see here. And what the difference between the standard Rosenblatt perceptron and that we propose here? Firstly, we have now a teacher who can say what is the target vector that we should use. Also, what kind of input is here? Really, the input may be various information from that is received from different areas of brain. It can be visual, auditory, somatosensory, and etc. kinds of information signals. And because we don't really know the exact vectors of inputs, so in our simulation we took as inputs vector just a set of random vectors x. And what is output, the target output vectors? Because we have now a teacher, so we suggested to use we interconnect these neurons and neurons in a way that I was described previously. That is, these N neurons create attractor neural networks with preformed attractor states. And when we show some input vector x, the signals from the input neurons propagate to the output neurons, and after that, due to the recurrent connections between these neurons and also due to the input signals, the network converges to a stable state to one of the attractors, and we used this rule, Rosenblatt rule, to teach the perceptron. And in such a way, after the learning, each grandma that is shown as the input will have the internal neural representation as an attractor. Here I would like to show you that really there are some theoretical constraints of the maximal capacity of such perceptron-like schemas. If each neuron is involved in k different attractors, and k is very small, much more less than the whole number of attractors, then the output representation will be very sparse. And due to this sparseness, it follows from the Gardner theory that there is some maximum value of the patterns that can be learned by such perceptron-like scheme, and it's estimated by this formula. And from this formula, it can be obtained the number of inputs neurons, R, and for the same values of the number of neurons, for the same value of activity and value of number of attractions up to million, k is equal about 1000, and we obtain that R must be no less than 12,000. This is theoretical constraint. And finally, I would like to show you that it's interesting that the proposed scheme of attractor-based perceptron has noise, very good noise immunity. What does it mean? When we if after the learning we show the same input patterns that we use during the learning, but with some noise, it is noise. When we increase the noise, nevertheless, the same attractor, the same grandma is firing. And without a learning, there is no noise immunity, and even for very small noise, the network will change their activity and will no noise resistant. And also here is shown by the red dots what will be if there are no recurrent connections in output layer, that is without attractor states. And you can see when the noise is very small, of course it's not so bad, but for bigger noises it's very bad. And let me conclude my presentation. The modern grandma neurons, according to their discoveries, are members of population of many neurons. And that might mean that there must be much fewer grandma cells than the number of neurons. And we propose a model that we demonstrate that the natural neural circuitry with inborn connection might have much more grandmas than the number of neurons. And also we demonstrate that the reverberation might extremely noise tolerant might make extremely noise-tolerant representation in neural systems. Thank you. Here I presented the model that can be used instead of this rule, the Heb rule, because the attractors in such way are formed without any learning. That is in embryogenesis. And there is no learning. These attractors are performed and then they are used as the ready structures of the brain. And here, these neurons is that attractors. And connections between them are not modifiable. But these connections, W, are modifiable. And they are adjusted in such a way that represent the input, various input in this stable representation. Is it? I'm sorry, I'm not understanding the question.
You know, there are two types of synapses between the neurons of the represented network, and they are unmodifiable. They are just inborn. And another is connections of the group with the external world, with receptors, and they are unmodifiable.
Yes, I have a question in relation to one of your past slides where you show noise resistance. I wonder, I understand it's not included in this presentation, but I would imagine that you was monitoring how this system resistance to noise improves
during training, there was no noise. And after learning,
But you could have thought as you did.
yes, I did it, but here it's not shown. Really, we saw how, if is it possible to learn on noisy patterns. Yes, yes, it works with little noise. It's very good works.
I'll ask more questions.
Yes, of course.
And then in your simulation, you found that with 50,000 neurons,
Uh-huh.
I don't see how you can get a failure calculation. You think that it's just something that you set.
K is equal to what? Yes, K is equal to N times L divided M, really. And here we can change the value of K, we can change the value of L. They are they are related. And here we just took the L equals to 50, and so K is very simple to calculate.
In the experiments that the CLA medical school did, in your case, you have overlap of the effects, right? When you have K over 1,000, that cell essentially encodes many different grandmothers, right? But they have tested a single cell and found a single set response only to like Jennifer Aniston,
Mm-hmm.
and not to other things.
Yes. I think that it have a very simple explanation, because in the experiments of Quiroga, Koch et al., they have a finite number of people, about 100, and they recorded the neurons, the finite number of units. So it's unlikely that one neuron will respond to several different peoples, because of the small number of the peoples and the units recorded. His theoretical model shows that it must be, but in the experiments it's not possible to show to a person about thousands different actors, and recorded all the brain. In human brain, it's not possible. Is it? So why not? Uh-huh. Uh-huh. It's an interesting question. I didn't think about it, but K equals to But I think that this estimation is not very bad. It's near the bound.
Okay, let's move to second talk. We might learn more to answer our questions during a talk from Celia. Thank you.
We have a problem in changing the presentation. There is something strange. Okay. First, thank you, Irina Belozova, for invitation. I'm pleased to present my first work in neuroscience here in Barrow Institute in Arizona, between saguaros and palms. And my name is Ksenia. I'll tell you about how neural networks can represent continuous values. So the signal of external world act on our receptors and is coded and processed at following levels. The mechanisms of this process is one of the most essential unsolved problem in neuroscience. One of the sub-problem in this field is continuous values representation, like an arm movement direction in Apostolos-Georgopoulos experiments. One of the well-known models of such things is a continuous or bump attractor. In this work, we focused on how does the network with continuous attractors can be created, and how does different points of it can obtain its values, as the movement direction, for example, using the signals of other levels. For the very beginning, I'd like to remind you one of the classical experiments. The rhesus monkey had to choose to which light diodes in one of 8 directions it should point to. At the same time, there were registered the activity of the neurons over the arm motor cortex area, and it was detected that the activity of the registered neurons have a bell-form curve with a maximum in a self-preferred direction. Also, it occurred that the population vector, which calculated by means of this formula, coincided with the vector of movement direction. Say iter is a preferred direction, and VE is a firing rate of the current cell. Alexander Lukashin is a Russian theoretician who worked with Apostolos Georgopoulos, and he elaborates several models for these experiments, providing the connection of neural activity and activity of the arm trajectory. One of these models was an attractor model. Now I'd like to define one of the most essential terms. I'd like to tell you about the simplest bump on continuous attractor network. So here and further we'll use the simplest model of binary neurons. And in this model, the stable state is just a set of neurons. The first model of continuous attractor was introduced by Sunichi Amari in 1974, and that looks almost like his model. So we put order numbers for all neurons. And each neuron is positively connected with his L neighbors in both sides. And it has inhibitory connections with the rest neurons. It's a Mexican hard connection pattern. If the negative connections are strong enough, any L successive neurons will be an attractor point. So it will be a stable state. For convenience, we used a ring attractor where the first and the last neurons are neighbors. Here you can see the example of matrix, of connection matrix in this case. T is a connection matrix. The next essential term is self-organizing map system. So one of the basic principles of neural system organization is keeping on relation of closeness at different levels of neural system, from receptors to cortex. The principle is somatotopy in proprioceptive system and tonototopy and rethinotopy in auditory and visual system. The first attempt to implement these principles in artificial neural network was made by Teivo Kohanin in 1982. He demonstrated the neuromorphic system, which made a representation of external input, external signals on a chain or on a lattice of nodes, which were very poor related to neurons. Here, I would like to demonstrate you a system which resembles to Kohanen's construction. However, in our system, the external signal is represented in neural network activity. Our system consists of two main parts. The first part is a recurrent network, which consists which includes continuous attractors, as was described before. The second part is a layer of air neurons. I call them receptors. Receptors are not connected with each other, but each receptor is connected with all neurons of the recurrent network. The essential thing is that a matrix T of connections in a recurrent network is constant. So it's like preformed. And the matrix of connections between the receptors and between the recurrent network is random at the beginning, and it changes during the learning process. The learning process consists of many repetitions of the following procedure. Receptors receive input from the external world and transfer their states on the to the neurons of the recurrent network. The activity of the recurrent network evolves in time in accordance with activity of receptors and other neurons in the recurrent network, and it comes to one of the stable state of this net. Afterwards, the connections between active neurons and between active receptors are changing dependent on their mutual activity states. This is one step of the learning procedure. The system behavior might be tested with frozen connections, so without point form. Later I can explain more details, but now I won't. So a few words about input states space. So an input signal has the following structure. The receptor's activity is set as a real numbers. So the vertical coordinate is a number from 0 to 1. And the representation of the input is a bell-form curve with the top in 1 point from 0 to R. For better visibility, the picture you can see the magnitudes of receptors excitation. So the vertical coordinate is for 3 input values. To show the modeling result, we used the following methods. First is the matrix W connections between receptors and neurons. In A, this matrix is random, and in B, that's the matrix after the learning process, and it looks essentially diagonal. On the horizontal axis, there are a number of neurons. On the vertical axis, there are a number of receptors, and color in the crosstalk means the value of the weight between the current receptors and neurons. At figure C, each row represents the state of activity of neural network for fixed values of external signal. The value of signal changes with lines, and blue is silent neurons, and these are active neurons. The ring diagram of the figure D denotes values of the external signal parameter in a ring, external parameter, and the odd number of the attractor states, external ring, which represent the input signal. The corresponding points are connected with lines. Figure B, C, E, D are given for the system which has completed learning process. Now I'd like to demonstrate 3 important properties of this system. First one, the property of scaling of representation of fragments of the whole range of the input signal representation you can read here. In cases when some ranges of values of external signals can be used with more probability than the other. So they mapped onto a larger number of attractors. We consider this property resembling the famous homunculus picture on the somatosensory map. Next, the increase of noise tolerance due to collateral connections. Figure A, B, E, C shows matrix W and mapping diagrams for different values of noise amplitude. Figure A and B represent a system with tolerable and intolerable level of noise. In case C, the noise was distributed inhomogeneously. over the signal space, including intolerable level of noise. What you see here, it occurred that the parts of the system with low level of noise helped the system to deal with parts with very big level of noise. And we have a good representation in our system. The next is the property of simple connectedness of mapping to the neural networks, the set of attractor points of which has more than one component of connectedness. That means that in our neural network with n neurons, there were not one but two continuous attractors. There are connection matrix for both of them in different numerations. That means that all stable states of the system belongs either to one ring or of states as described earlier, or to another state, which differs from the first state by the enumeration of neurons. When this network was exposed to the learning process, finally we observed that the mapping converges to the state where external signal is represented with activity of one of the rings. The choice of the ring was due to spontaneous break of symmetry. In the first part of our work, we didn't discuss any possible mechanisms of such network creation, of continuous attractor emergence, and also we didn't discuss the biggest possible length of a continuous attractor in a network. In following section, we are going to do that. So that's a molecular marker model. It looks like in Yakov's talk, but it's has different. We consider here that connections in the network are determined with the help of special markers. Furthermore, we consider that markers have different degree of tolerance to each other. They like those who has similar color and dislike those who have very different color. And let us have markers all of different colors, and suppose that we can arrange them with continuous transitions of colors. To compose the network, we distribute the markers between the neurons randomly, except the fact that one neuron can get only markers which are which strongly dislike each other. Afterwards, we connect neurons with markers who like each other. For example, in some degree D. For example, for this emphasized neuron, when D equal 1, he has connections with these two markers because of yellow marker, and with these two markers because of blue marker. Here a diagram which illustrates the formation of network. They can be different attractor states, and each neuron can be included in different attractor states. And now we decided that we created the network with continuous attractor. But of course, we should check if there is a continuous attractor in our network. And there can be different methods of checking the attractor states in the network, but we used the dynamical methods. The main idea of a dynamical method is that we add one more mechanism in the dynamics of the network, something like calcium-dependent potassium channels. So when the neuron is active during some time, his threshold grows up. And this value is increased for all neurons exponentially. But in case we don't know what are the stable states in our network, the only thing we can do is to compare states of the network in different moments with each other. That's the main idea of L-plot. In horizontal axis, you can see time, and in vertical axis, you can see the time difference between the current states and the past states and the future states. The color means the difference between the distance between the states, the current states, and the other states. For example, the blue means equal states or zero distance, and 30 orange means that the distance between two states is 30. So two states have 30 different states of neurons. Parallel blue lines means that the activity in this system is repeated with some period. The other way to find the continuous attractor is have the same principles as L-plot, but I called it absolute dynamical method. We can use it, for example, when we can detect all the stable states, and we compare the state of the system with stable states we know. So in horizontal line, there are numbers of stable states we know. In vertical axis, we can see time. So the activity goes through all the stable states we defined. And in such case, when we have M markers and we can detect the changing of all stable states without some jumps and interruptions, we say that in this network there is a continuous attractor with length M. And now some properties of such attractors. Applying the described absolute dynamical method, we model the molecular marker networks with different M and N. In this case, N is 300 and M is N, 2, N, 3, and so on. And on the top picture, there are plots for values of thresholds, of fatigue for all M attractors. At a certain moment, it looks like a kind of wave. And below, you can see the absolute dynamical plots. For K from 1 to 6, we could get a continuous regeneration of an entire continuous attractor. So we observed it. And for K7 and 8, we have observed jumps, a lot of jumps, from one attractor point to another. So we assume that the maximal length of continuous attractor is 6n, for n equal 300. And we repeated this test for n 600, 900, and 1,200, and indicated that the maximal attractor lengths will be 11n, 17n, and 23n. And it's good approximated at a straight line. If we get, for example, n equals 50,000, as in cortical column, k could be 1,000, or the length of the continuous attractor is about 50 million states. And conclusion. We have developed a neural network system which can detect one-dimensional set of signals in the input signal space. We have explored the possible number of stable states in the neural network which can be used for the first task, and have explored a possible method of synthesis of such network. And the present work, in present work we used networks of formal neurons or binary neurons, but we have got the initial data that the same result can be obtained for Leaky integrated fire neuron model. So, thank you. That's all. Your questions? I think that these properties that we explored in our work for formal neurons, for the simplest model of neurons, they can be obtained in more complex models on neurons, because it's much more easy to have every properties when we have complex models.
Okay. Everybody is here? You told me, yes. I know. Really? Thank you. Yes, this is really a great honor to talk at one of your seminar sessions and to have possibility to show you our initial results. It might be too brave project, and to share with you our hopes. Please take our sincere gratitude, and first of all, they are to my longtime friend and collaborator Irina Beloozerova, and also to department chair Dr. Alan Gibson, and to the beautiful Mrs. Mary Fifield for making that possible. Yeah. So I hope that most of you at least try to read our claims at that website. And if so, I'm ready to answer your questions, but after the talk. To be honest, the project does not look like an ordinary or even extraordinary project in the field and meets storms of critics. Our talks today are not intended to defend the project ideas or express, but just to show our initial results. The true job will start at January 2013, but we are in a low gear since June 2011, and we are going to show you preliminary results. And questions may be answered after the talk or maybe even tomorrow if anybody wants that. The project is David Marr Memorial Laboratory for Brain Reverse Engineering. David Marr lived in those years, and he, in 3 years, from 1969 to 1971, he has published 3 papers of how cerebellum, neocortex, and hippocampus work, and many of ideas of his work were turned to be true. And these 3 papers were published by Lucia Vaina in 11 years after David's death. But the initial idea to get together these papers was, here it is expressed in David's letter to me. He agrees to publish that in Russian. Unfortunately, in Russian we didn't get it published. The letter is of 73. So, but my specific talk today is about the cerebellum. I will tell you about a concrete piece of knowledge which we probably added here. Besides, I will try to present a broader picture of cerebello-neural mechanism. What we added, it's just a concrete models in the style of Marr, Olbus, Ita, Mauk, Schweihofer. And it just models something like how a child learns to follow an object. And in this work we first found that a simple model of the cerebellum can do something like that, and second we have found something more special which I will tell in proper time. So now I will start to proceed in the detail. I will not talk about the cerebellar physiology because I suppose that audience is familiar with it. I just stress on some particular issues which are important for this concrete model. Yeah, the most beautiful thing in the cerebellum is Purkinje cell, and in human it has 200,000 parallel fibers coming to it, and exactly 1 climbing fiber, which wields probably several hundred synaptic contacts with Purkinje cells. The synapse between the climbing fiber and Purkinje cell is the most strong synaptic contacts in human brain. However, it has some properties which make this contact really strange, even enigmatic. Climbing fiber fires with a frequency of approximately 1 in a second. Each impulse of climbing fiber elicits impulse or 2 or 3 of Purkinje cells, so it's very powerful. But each climbing fiber, besides going to Purkinje cell, it goes to nucleus and provides just excitation straight to nucleus and inhibition from Purkinje cells. So virtually it's just a zero effect of climbing fiber over the downstream neurons. So this most powerful synapse as if does almost non-job. But the impulses of climbing fiber are very important to Purkinje cell. You know, of course, that in recent and in now just published new work of Alan Gibson, they showed that just destroying, stopping the impulses to Purkinje cells dramatically changed the whole picture of work of the cerebellum. This need activity, the Purkinje cell need the activity of climbing fiber. Then also it is known that as a consequence of climbing fiber action, there is a long-term depression on synapses from granule cells. And also the fact that stopping the climbing fiber activity imposes the increase of activity of Purkinje cells means that in absence of climbing fiber impulses, the connections between parallel fiber and Purkinje cell increase. So Purkinje cell need both activity and inactivity of climbing fiber. Then in the cerebellum recently it has been discovered a very unique closed loop. The closed loop consists of Purkinje cell, then of inhibitory neurons of nucleus, of cerebellum nuclei, inferior olivary cells, or sometimes I call it liana cell because climbing fiber is a kind of liana which goes to a Purkinje cell. So, and the liana cell goes exactly to the same Purkinje cell which acts on the nuclear cells. So this is a closed loop. And formally, this is a positive feedback because this is an excitatory connection. This connection is inhibitory, and this is also inhibitory. Inhibitory multiplied by the inhibitory means excitatory connection. That's the loop. And this loop, I will show some features which this loop can have. It's just the same drawn in another way. Probably I will use that to explain what can go in this, what is going on in the system. So let's consider that all these signals are constant, and these signals are constant. If they are both constant, then very soon the Purkinje cell comes to the state when connections between granular cells and Purkinje cells are changing, and so the system comes to the stable state. And in this stable state, what kind of change of connections in this system. Here is just the rate of changing of connection weights of granular cells to Purkinje cells. It depends on two factors. One factor is just activity of climbing fiber of liana cell. And another factor is activity of granular cells. And it is known that for close to moment of generation of complex spike, there is a long time depression and long time potentiation all the rest of the time. And there is such a time interval, which may be called equilibrium time interval, when depression is equal to potentiation. And this is exactly the period with which usually Purkinje cell fires. And as soon as you will put constant signals on the both inputs, on the granular cell inputs and on liana cell inputs, very soon the system comes to the stable state. However, here is the intercellular potential, or model intercellular potential of the liana cell. You know, the cell continues to fire in a random pattern. The whole system stays in the stable state, and there is approximately constant frequency of Purkinje cells, but this cell fires in a random pattern. And here is, for example, this is a pattern, just time interval, just sequential time intervals for real Purkinje cell, for complex spike of real Purkinje cell, here's 3 seconds. And for model cell, believe me that their resemblance is really unique. Any other cell which I tried to draw picture with the other cells with the same part, they are not like what we can get from the model, or which is what gets from a real Purkinje cell. So it's a kind of dynamic chaotic system. And also, what happened if you have just different, here is, let us denote alpha is a constant signal which comes to liana cell, and beta is a signal which comes from nuclear cells of the cerebellum, which are due to Purkinje cell activity. With different alpha and beta, always such a rule is performed with constant in time alpha and beta. And even if they vary in time, then they working just compensate each other. It's like yeah, here. I tried to draw the membrane potential of climbing fiber cell consisting of 2 parts. This part consists of red input to the cell from this part consists from one input to the cell. And this part consists from the other input to the cell. And they all compensate each other. So that finally the membrane potential is the same as for constant alpha and beta. But in some cases, in this case, Purkinje cell activity goes ahead of that activity which was on the input. So that is the properties of that system, which can be described by not complicated equations. But what is important in this system also, that you should probably use here pointing how the connections are changing, the factor of climbing fiber, and also not exactly activities of granule cells, but activities transformed by metabotropic receptors. So it's much more, so the changing of connections probably is due to the metabotropic factors, not just straight to the receptors. And if here we made such a, this is a model, and in this model, this is the input signal to Liana cell, and this is membrane potential of Liana cell in initial stages of exposure and in later states of exposure. At later states, the input to Liana cell is still periodic, but the total membrane potential is straight. And if you look at these are periodic signal, we can just show the histogram of firing of complex spikes during the phases of periodic process. And in this stage, the firing is here. And in this phase, the firing is all over the phases of the periodic process. So that's what the system can do. And here we tried to apply this system just for very simple, I would say even toy problem. It is just a model muscle, kind of a model muscle which rotates an eye. And there is an object on the screen. And just the signals from retina goes to as mossy fiber to Purkinje cells. And they also go as a climbing fiber, as input to climbing fiber cell, here, to Liana cell. And initially, you have no specific connections of Purkinje cell to anything. And here is what's going on in the system. It's the initial state of the system. Here are movements of the object on the screen. And here are the eye direction. And in the beginning, it's, of course, in the first phase, it just eye is randomly wandering over the screen. And then after some exposure, it goes to such a beautiful picture where this is the movement of an object. And here are impulses of climbing fiber. So we were just interested in two things in this modeling. First of all, we have obtained that this simple construction can do useful job. It really, of course, the real system for eye tracking is more complicated, but the principle, of course, will work even in more complicated system, but it works very good. But what surprised me, at least, that still there are a modulation of complex spikes. While everything is learned, the complex spikes are modulated. I showed you in the previous case that when anything is learned, there is no modulation. There is just random scattering of complex spikes. But I just remembered that there were results of Simpson and co-authors, where they have such, at least pictures, when in some directions they have something, a rabbit and rotating external visual world, and there was a case where there were impulses in one direction, and there was no complex spikes in one direction, and no impulses in opposite direction. So that reminds me, here, for example, the perfect. What we got here reminded me of that case. And as a probably a very fine, the model. I just claim, I don't claim that our model is exactly a model of this physiological process. But I claim that probably the persistent firing of climbing complex spikes in one phase and absence of them, it has something to do with our model. And as an additional evidence of that, when we looked at really pictures there, it's clear that in phases when there are complex spikes, they are twice often than in background. In background, probably, we have this equilibrium distance between complex spikes. And when we have such a case, when the system is in equilibrium state, but there are impulses only in one half, still there are these impulses, they have twice as large frequency. And you can see that it's probably, again, the stability. Each granular cell weight, it grows when there is no complex spike and decreases when there is. And so there is a stability which consists of small increase, small decrease. So each granular cell has in average constant weight, but in general they can do something. We don't know what they do here in rabbit, but we know what they can do in this model. So that's probably two things which I concretely can tell you which we got in the cerebellum in the recently. But now I'll try to go to some unsolved problem maybe in cerebellum. Of course, you know that cerebellum is 10% of brain weight and from 70 to 90% of neuron count. And so there are many unsolved problems here. And of course we can't answer all of them, but I will just stop at four concrete subjects. The first one is a problem of electrical connections between inferior olivary cells. The second will be just what is the sense of the signal which comes to inferior olivary cell? What triggers inferior olivary cell and how it can be used? The third question is about the neuromodulators. You know that in the cerebellum there are at least extremely well expressed biological amines and also nitric oxide and cannabinoids. And the last question is, you know, the mechanisms of cerebellum are very fast changing. You know, in those experiments, say, of Allen, the change in behavior starts in 10 minutes after you have made injection. So anything in cerebellum changes very fast. But if the cerebellum can be used for long-term learning, like learning to use standard shift or not, I don't know. So that are the question. Then to the first of the questions, it's about the gap junction connections between cells in inferior leaves. You know that, I don't know if exactly Rodolfo Linus has discovered these gap junctions, but at least he was close to the discovery of that. And immediately as soon as they have discovered that, they claimed there are synchronicity between different climbing fiber cells, between different complex spikes. And this synchronicity is provided with this gap junction, and it is useful for. However, many people, unlike for many other discoveries of Rodolfo Linus, this discovery hasn't been confirmed because those who looked at activity of in inferior leaves never saw the synchronous activity of the cells there. And here's a very interesting finding. It's a theoretical finding. Here, what you see here is just model inferior olivary cells. There are exactly 20 inferior olivary cells, and they get exactly the same input signal. And in this case, you can see that all 20 cells are just synchronous. And in this case, the gap junction or electrical coupling coefficient between cells is 0. Because cells get the same, they are synchronous. But if you will introduce connections between cells, then again with the same signal input, you will have asynchronous activity of these inferior olivary cells. So it seems that really, and of course, you know, as climbing fiber activity determines finally which granule cells and how they are connected to their Purkinje cells, making them all the inferior olivary cells synchronous make no sense at all, because it just lose of hardware. And here we can we have a hope that hardware is not lost. It can be effectively used. So that's what about the biological amines. You know, this question is still very far from hopes to be solved soon, but the hope is that there are with some way control the rate of learning in the cerebellum, or the rate of the plasticity processes in the cerebellum. And might be they can stop the plasticity in some places, or something like that. For example, you know, one of the most traumatic issues which get astronauts coming to Earth after the long-term journey is connected to cerebellum. It might be that they can be reduced if we will temporarily stop the learning processes in the cerebellum. I don't know, and then we can use the system. It's just speculation and almost ungrounded. Yeah, and the rest, the last problem is also how, if we can acquire permanent, if we can get permanent learning in the cerebellum, like learning to ride bike, or we can't, it probably also depends on the previous problem, but still I have no general idea which will explain how it works, because obviously in any part of the cerebellum, stopping the climbing fiber dramatically changes everything, and that means that the process is going on all the time. That's it, and now probably just I will express some optimistic point of view. I am in the field for 50 years, and in the beginning we have only questions, and we have infinite number of questions. We didn't know even what to ask. But now, 50 years afterwards, we still have huge number of questions. But it's probably a limited number, huge but limited. And that may give some hope. So, so what I want to tell here is a picture of our initialization. Here is a picture of what we are doing, and here is the picture of our initialization meeting in 2011 in Sudak, and he is my co-author in this talk, and no other of team is present here. These guys are younger, they came later, and thank you. What? Yeah, yeah. Yeah, it's just about how that there could be such a state when still complex spikes are synchronous with dynamic activity, but the system resides in a stable state. There could be such examples. And moreover, I would just add here that the idea that climbing fiber just tells about errors, it's probably not exactly true. And what is more true is that Purkinje cell repeats whatever it has got to the climbing fiber cell. But this repetition is not a plain one. It's very creative, as to say. Yeah.
The Gibson lab has showed in several publications that during an active movement, that activity in the periodicity can be suppressed to some extent.
Yeah.
How does that finding fit with your calculation?
Yeah. Of course, I should look at the data just to look for how long it holds breath, I would say, because it seems that Liana cell should breathe all the time with this 1 per second, and of course it can hold for a while, and when it holds for a while, it denotes that the synapses from the active granule cells on Purkinje cells are increasing their weight. And probably in many active movements, this is the case. It's
Is there any study like that in retina?
where?
In the retina?
In retina? Now, I was telling about this specific pattern or specific network of cerebellum, and the retina of my model is very far from what is known in real data. Why I showed the Simpson data? Because there in this rabbit experimental model, it's very much reduced experimental model, and he claims that he works with a force, if I'm not mistaken, force visual system, which is very simple, and which is probably the same as we use. Just seeing is the object move or doesn't move or on the retina. That's all. If the image on the retina is stable or slowly or not slowly moves. That's the only thing. Yes. Yes. Yes, you see, the LTD is very well known. And the LTP, in fact, it is demonstrated in the experiments like the Allen's. And the first experiment of this type, yesterday we talked a lot with Alan about this stuff, and of course we with him know that the first experiments were of Colin, Manil, and Disclean. It was in 1980, when they found that as soon as you stop activity of climbing fiber, immediately you have increase of activity of Purkinje cell, and the process goes wild in 10 minutes. So there is a 10 minutes when, and as I know, nobody directly showed that this is due to this process, this active granular cells increase their weight. But probably somebody did it. I don't know exactly. Yeah. Plasticity is going on all the time. The only idea of this guy, Schweihofer, is that the modulators can change the rate of plasticity. But plasticity goes all the time. It goes positive in absence of complex spikes and negative in presence. So it's dynamic. That's one of the reasons why I told that it's not yet clear how we can acquire really good skills, motor skills with cerebellum. Probably we can, but it's an enigma how it can be done if, and probably this is the proper picture of what is going on there.
Are you trying to demonstrate that the Purkinje cell is a motion detector cell? Is that what you're trying to simulate?
No. The Purkinje cell, activity of Purkinje cell, it's very rarely, I know only one example. I don't know if I have it here in this demonstration or not. I know only one example where there is a very strong correlation between activity of Purkinje cell and something outside. Usually, as Purkinje cell acts upon, probably, I would just, I don't know exactly, Baev can tell more about that. So probably Purkinje cell just is an input to CPG or pattern generators or downstream from cerebellum. Or maybe, you know, for recently, almost 20 years, it is known that cerebellum is involved in cognitive processes. And you know, as to involvement of cerebellum in movements, it was known, yes, it is involved, however, you can't clearly describe how it is involved. You can? You can?
I can understand. During locomotion, Purkinje cells are clearly and very profoundly modulated in electrical stimulus.
Sorry.
It has been shown in 80s by David Armstrong.
Great. I should read literature. Thank you. I should, but how about the complex spike modulation? Was it?
There is no complex spike modulation.
Yeah, great. That shows that my picture is right.
But the major output is going to be the simple spike activity, which is going from nuclear.
Yes. But yeah, sure. But this output is just sculptured by previous action of climbing fibers. That's the explanation. While you have no modulation of complex pi, because complex Liana cell have a flat membrane potential, it is.
But you were showing us Jerry Simpson's work there, showing that complex spike activity does modulate with whole field visual optokinetic stimulation.
Yes. This is what I showed here. But I just demonstrated that there were
Then to follow up on that, Jerry Simpson's work has since shown there that what happening is associated with retinal slip. That those cells in the uvula of the rabbit are responding to vestibular stimulation.
Yeah.
When you paralyze these little guys and you have the eyes continually slipping, that's what's causing the fire.
Yeah.
So that does relate to the activity.
Yeah. Thank you much.
I always have a question. Is it known why simple spikes are depressed after complex spike?
Now, first of all, inactivation definitely happens there because you have such a strong depolarizing event. And probably it's just an activation of sodium channel threshold.
It's more related to calcium channels with the climbing fibers. So it's a calcium channel.
Yeah, but still, why is this pause? The pause not always happens. It's probably 10% of complex spikes are not, they have no complex spike pause. No, you see, I was just very pleased to see some other modeler who independently of me decided that he should not care about the action of climbing fiber on output, that they are canceling each other. But with the pause, of course, this pause is not cancelled, and I don't know its function. And of course, it's probably one of the puzzles, one of the problems is the understanding cerebellum.
I'm not an expert in neuroscience, but my sense is that motion detection actually occurs before in the visual cortex. Is that true? Motion detection.
It might be, but it's out of scope of my talk. It's just now I am very narrow here, so I don't go. And this Jeff Simpson model, they have no cortex at all, I guess. And
They do, because the activity is coming down through the mossy fibers that are coming down to
they do, yeah, might be,
visual cortex.
yeah.
Initial activity.
Yeah.
That's being fed down to the pontine.
But he just destroyed something. I forgot. This preparation is very much reduced.
Destroying the otoliths and the semicircular canal.
Yeah. Thank you.